first like to thank the organizers for the invitation to speak today. It's a great pleasure to be able to present to you some of the work. I started my lab in about July, so it's been a really trial by fire and a real exciting opportunity to really see how we can, in a biological engineering department, really try to think about the critical problem of tuberculosis. And before I dive into my talk, what I want to show you is what you're looking at here. So here are just macrophages infected with kind of a transcriptional reporter strain of MTB viability. And what I want you to take away from this is in blue are the nuclei, and red are what we call transcriptionally inactive bacteria, and in green you see what we call transcriptionally active bacteria. And as you can see from this slide, and I think what has become really apparent in TB, is that there's a tremendous heterogeneity in kind of single cell responses at the level of the bacterium, at the level of the macrophage, and as we begin to build up to larger kind of immune ecosystems such as the granuloma. So I'm just going to go ahead and dive right in. So I think, just as a reminder, I think it's really important to remember that tuberculosis is a huge global health problem. It's estimated that 1 in 3 individuals globally is infected with a bacterium, so that means about kind of a disease load globally of about 2 billion people. And one of the things that makes TB really, really difficult to treat in these human populations is an incredibly variable course of infection in infected individuals. And what do I mean by that? So the kind of the global paradigm that has existed for a long time in the field is that you have 1 in 3 individuals who are infected, but about 10% of those individuals go on to develop what we call active disease, which is characterized by your classical symptoms: coughing up blood, weight loss, fatigue. 90% are virtually asymptomatic or latent. Now, just as a reminder, while in the last talk we saw really great examples of immunotherapy, vaccine development, we currently still do not have a protective vaccine for tuberculosis. And the questions that we try to answer in our lab are, what does immune success look like, and how do we get there? And so one way that the field for a long time has thought about how do you actually understand what's different and what contributes to TB control is to ask what's different between those individuals who develop active disease relative to those who develop latent disease. And this is what we'll characterize as kind of a global model. There are kind of global immunological features that distinguish between kind of latent and active disease. And so you might set up that model, but actually over the last maybe 5 or 6 years, what we've actually begun to learn is that TB, instead of being kind of globally determined, is incredibly geographic in its nature. And so what do I mean by that? So here I'm actually summarizing about 7 years and millions of dollars of research in one simple slide. And this is work largely done first in the non-human primate model to develop the tools, but now being done in human studies where you can, for example, infect non-human primates with molecularly barcoded strains of MTB and kind of watch the dynamics of inflammation using PET-CT imaging. So in the non-human primate, what people have begun to observe is that the disease is incredibly highly geographic. So you have these individual foci of disease, otherwise known as granulomas. And if you actually monitor kind of dynamic inflammatory trajectories in the lung, if you look at the PET signal in this lesion over time versus the PET signal in this lesion, what you observe is that these lesions kind of have kind of unique immunological trajectories in terms of their inflammatory response. Now, if you actually just take those lesions and then profile the T cells in lesion A are very different functionally than the lesions in the T cells in lesion B. And then most important is if you actually take these individual lesions and actually do kind of deep sequencing of the bacteria, what you observe is that by and large these lesions are founded by a single bacterium. So now if we zoom back in time, what this means is that really the kind of the nidus of infection, what starts it all is an interaction between a single bacterium and a single macrophage. And that's kind of what as engineers, we like to think of it as the kind of the initial conditions that set up the entire system. And I think the most important feature that I really want to highlight for you here is that if you actually take these lesions and plate for viable bacteria by colony-forming unit assay, and this is really important, so independent of clinical status, some lesions can fully sterilize, whereas some lesions do not. So this means that immune success is possible, but the question is how do we get there? And I think for the sake of time, I'll just skip through this slide. And so the questions that we ask in my lab are who are the cellular players and what really contributes to, what are the molecular drivers of immune success? And like I said, one of the questions, the way that we like to think about this problem is how are these initial conditions really established in disease? And so the way that we do this is we decided that if we think about it, one of the kind of the critical things you might think about, how do you actually control bacteria? So I think while the field has spent a lot of time thinking about T-cell responses in TB, what I think we really want to begin to think about is if we want to achieve sterilization, it's really about how do you actually drive the infected cells, namely macrophages, dendritic cells, and neutrophils, to bacterial control. And so those are the questions that we ask, and we've by and large started with the question of how do the macrophages actually contribute to both variable granuloma fate, and how do we actually manipulate macrophage function in TB disease? And so when we started this work, we started with this simple model where, for example, you might imagine that you want to move your macrophages from a kind of a permissive state, one that permits bacterial growth, to one that's restrictive. And I think we have lots of different models for thinking about macrophage states in disease. And what I'm hoping to tell you about today is a really kind of exciting story that has been developing in our lab to really understand how do you actually move along this kind of immunological trajectory, so to speak. And so I think it goes without saying that the kind of the paradigm in the field for a long time has been that you can start with kind of a neutral macrophage state, let's just call it M0, and we have these two kind of phenomenological extremes, kind of hyper-inflammatory or immunoregulatory, and that the model is that these cells are kind of incredibly plastic and polarizable. And so the work of a really talented medical student who unfortunately went back to medical school last week is to really begin to try to understand more complexity in TB infection and TB infected macrophages. Instead of just using two models of macrophages, what we started to do in our lab is build libraries of different macrophage phenotypes. And so the way that this is done is we're a human macrophage biology lab, and I want to underscore that. So by and large, what we do is we work with healthy CD14 monocytes that we get from healthy donors. We differentiate these macrophages using M-CSF or GM-CSF for 3 days. Then we replenish the media and add both M-CSF and additional range of different cytokines, and that's what I'm showing you here. And then on day 7, we started to just ask about the functional properties of these cells, their ability to respond to TLR ligands, their ability to control TB, to really begin to build kind of an enhanced molecular characterization of these cells. And so one thing I've always been envious of T-cell biologists is when they show FACS plots and they say, I have double positive this, and I can identify what kind of T-cell state that I'm in. But I think anybody who's ever tried to analyze macrophages by flow cytometry has always run into this problem that as you compare these different macrophage states, it's kind of quantitative differences as opposed to qualitative differences. And that's what I'm showing you here. So if we just look at the M-CSF cells and we look at a number of different surface markers, what you can see is that yes, there are differences, they change, they go from high to low, but if you actually use an isotype control and ask what % positive are these different macrophages, they're all positive for anything. So if anybody ever gave me a random tube of macrophages and said, what state are my cells in, I'd never be able to tell them because, in fact, this kind of binary gating strategy doesn't really work using the canonical markers that we have. So in an effort to really try to begin to understand, can we actually characterize these cells in new ways, what we started to use is single-cell RNA sequencing. And so just to even begin to look at the difference between macrophages derived with either M-CSF versus GM-CSF, we perform single-cell RNA sequencing and we can begin to identify a number of new markers. And so these violin plots are just giving you a summary of the single-cell RNA sequencing data, but really we wanted to know, could we actually translate any of these genes into new markers? And what I'm showing you here is now just some of this expression analysis by Western blotting. So one of the genes that was highly enriched in our GM-CSF-derived macrophages was this gene thyroidoxin, which we think is really telling us something about the kind of oxidative state of the cells. And you can see that our single-cell RNA sequencing did a pretty good job of identifying a new marker. Now, it's not showing up super well here. We actually identified another marker. This is ALOX5AP, otherwise known as FLAP, which is really responsible for the production of inflammatory lipids. And you can see that also marks GM-CSF cells. And really importantly, I want to emphasize that these are not just markers, I think they're telling us something about the actual biological properties of these cells. So actually work by Charlie Sirhan's group last year actually demonstrated that if you take these different macrophages and actually challenge them with E. coli, their ability to produce either the pro-resolving lipids or the inflammatory lipids was incredibly different. So actually just this simple difference in expression can really even set up kind of very distinct kind of local environments, especially now thinking about new axes in addition to cytokines, thinking about lipids. Okay, so this is really great. So we found some new markers. But one of the other things that we ended up doing in this characterization is we also performed a number of different ELISAs to look at kind of the soluble response in response to different TLR ligands. And what you can see here is as you begin to look maybe at the ability to produce particular cytokines, maybe we're better able to resolve these different kind of macrophage states. Well, one of the things that I'm going to really focus the rest of the talk on was this observation that was made when we looked at the GM-CSF derived cells. And, you know, I think as a new PI, you kind of learn by mistake sometimes. And I remember when my student came to me, and as you can see here, in the GM-CSF case, we really don't see any production of IL-10. So my student first came to me and said, I don't think I saw any IL-10. I said, I think you did it wrong. And, and I think that was a mistake because he said, you can do the experiment next time. And so for the next subsequent 5 replicates, I subsequently ate my words on 5 different occasions. And so what I want to really emphasize here is that not only are we generating, just one type of GM-CSF-derived macrophage, we're actually generating 6 different types and challenge them with a whole variety of ligands. So if you think about that combinatorial space that we're experiencing, this is a really robust phenotype. And so we said, could we actually use IL-10 as a prototype to understand this question of plasticity? So now that we have a marker that really kind of behaves as kind of this digital marker, can we actually begin to explore those dynamics? And so that's what we did. So we said, could we actually use this IL-10 secretion as a prototype for looking at these polarized dynamics? And so one way we began to do this is we actually took the GM-CSF cells or the M-CSF cells, differentiated them for 6 days, and then subsequently gave them either no additional cytokine, the same cytokine, or the opposing ligand. And then challenged with LPS and looked at IL-10 release. And here's something that you might predict. So if we start with the M-CSF cells for 6 days and then nothing for 3 days, you see this really characteristic response that you would expect. You see IL-10 in response. Now, if you just resupplement with M-CSF again, you don't really see a huge impact, but once you add GM-CSF for 3 days, then you can actually see that this IL-10 release starts to go down. Now, really strikingly, when we go the opposite way, what we find is that, at least for the ability to produce IL-10, these cells seem to be trapped in this kind of very stable phenotype. And one explanation for this is that there's just differences in receptor expression, but we actually characterize the expression of the receptors on these cells, and they're the same, suggesting that there is some other kind of mechanism that may be contributing to these kind of durable states that we're observing. And so you might have to say, in vivo, there's actually a whole host of different cytokines that the cells are experiencing. And so now if we actually begin to create a more complex model, where what we did here is we actually pre-mixed GM-CSF or M-CSF in escalating concentrations. Then we layered the monocytes on top so there was no temporal asynchrony. We let the cells differentiate for 6 days and then challenge with LPS. And so in the results, what I'm showing you here is that if you actually look independent of any concentration you try, if you don't add LPS, you don't really see IL-10. But as you titrate the amount of M-CSF, you can see this really nice dose response up to a particular point. Now, if we look at the GM-CSF case, no dose that we've tried has actually allowed us to see IL-10 production. Now, the question is, what happens when you combine them and you actually begin to look across the axis? Really strikingly, and I was flummoxed when we first saw this, that even if you go to 50 nanograms per mil M-CSF compared to 0.5 nanograms per mil GM-CSF, the ability to produce IL-10 was really dramatically diminished. And so we think that what's happening, at least in this case, is that GM-CSF is really providing a particular set of signals that seems really potent, and the question is, what are those signals? So if you spend a lot of time in macrophage biology, you know that if you just give GM-CSF to CD14 monocytes, people like to call those another model of generating an M1 macrophage. And so we wanted to ask, could we actually look at other models of M1 macrophages and try to explore these polarization dynamics? So that's what we did here. So we went with your very classical bread and butter model here, where we're going to differentiate our monocytes with M-CSF for 6 days, then add LPS plus gamma to make an M1 macrophage, or IL-4 to make an M2 macrophage. And the question we wanted to ask is, could we observe a similar kind of lack of polarization dynamic from the inflammatory state to another? And so in order to do this, what we just did is we did canonical flow cytometry. And what I'm showing you here is that if you look, say, for example, at CD206, which is a classical M2 marker, if you add IL-4, you see the characteristic increase in CD206 as you go from an M0 cell to an M2 cell. Now, really strikingly, if you start from an M1 cell and then give IL-4, you lose that ability to induce CD206. We can look at this across a number of different markers, and the trends are all the same. So again, if you look at something like CD200R, you no longer see this increase in expression once you start from an inflammatory macrophage position. Now, this has actually also been observed in mice, which is nice to sometimes have a phenotype that communicates between both mice and humans. Unfortunately, the mechanism that they implicate in this model is one involving inducible nitric oxide synthase. And so they were able to observe a very similar phenotype that they could begin to disrupt by using 1400W, which is an inducible nitric oxide synthase inhibitor. And really strikingly, if you actually look in their discussion, they say, although INOS inhibition clearly improved, blah, 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 it partially restores, except human macrophages don't secrete NO, thus unidentified mechanisms are clearly at play. And so we're still working out what we think those mechanisms are. We have a really promising lead that suggests that there's a transcriptional repressor that's actually becoming activated in inflammatory macrophages, which is actually blocking the expression of these classical M2 markers when you're going from this state. So what we think we're at least observing right now is that there's this M1 star, which kind of represents this kind of hybrid state, which may still have the ability to respond to IL-4, but not through the classical phenotypes or markers that you would typically begin to explore. Now, one of the things that makes macrophage biology even more complicated is the sources of your different monocytes. And so just as kind of a monocyte biology 101, just to remind you that there's actually, we think that there are 3 different flavors at the very least of different monocytes. Our classical monocytes, which is what all these experiments were done with previously. We have an intermediate monocyte phenotype and a kind of a non-classical monocyte phenotype, which are just observed by looking at CD14 versus CD16 expression. And so the question that a graduate student, Josh Peters, in the lab has begun to ask is, how does the ontogeny of these different macrophages contribute to their kind of functional properties subsequently? And this is really important because if we actually think about TB disease, these non-classical monocytes are actually expanded in disease. And so one of the, in active disease in particular, and so one of the questions we wanted to begin to ask is, how do these different kind of monocyte expansion dynamics, cellular dynamics, contribute to kind of macrophage function in disease? I'm just going to give you a little teaser for what that looks like. And so here what Josh did is he did 12 populations of macrophages by single-cell RNA sequencing and then tried to understand how do these different macrophages respond to various TLR ligands. And really what I want to draw your attention to is that independent of kind of the subsequent cytokines that are used to differentiate these macrophages, non-classical monocyte-derived macrophages actually retain a highly inflammatory properties. And so the question is, what really governs what makes non-classical monocytes incredibly inflammatory? And if you think about what that means for TB disease, we're actually, one of the axes you really want to think about is both managing inflammation and tissue damage. How could non-classical monocyte-derived macrophages actually be contributing to that axis? So what I told you at the beginning of this talk is that, okay, we've got this really complex molecular ecosystem of a granuloma where you have lots of different macrophage cell types, and we were going to do a very simple reductionist model where we take macrophages, infect them with TB, treat them with different cytokines, and hope that that's going to allow us to derive insight about what's going on in vivo. And so how do we actually first establish what that in vivo representation looks like? And so work that we've been doing in collaboration with the Shalick Lab, the Fortune Lab, and the Flynn Labs, the Flynn Lab actually developed the non-human primate model of kind of TB, is actually performing single-cell RNA sequencing on non-human primate granulomas. And so this is an unprecedented effort because it's taken a lot of effort, about 4 years, to be able to build out these technologies such that they're transferable and workable in a biosafety level 3 environment. And really what I want to draw your attention to is, while I spent the first half of the talk talking about 2 different macrophage populations, what we know is happening in vivo is that there's a much more complex spectrum of different macrophages. So this is just kind of all of the data in aggregate. This is what we call our macrophages, and if we actually just look at gene expression in a heat map form across these different macrophages, what I really want to draw your attention to is that the macrophages are very different. And so the question that we want to answer now is, how do we actually understand single-cell RNA sequencing data in a better context? And so, the theme of this session is concept to clinic. So how do we actually take, in vitro data, in vitro single-cell RNA sequencing data, where we can generate so many different perturbations and understand what that means about what's happening in a person? So the questions we wanted to ask here are, what are the cell states that may be shared between different diseases, and what genes might be signs of a larger contextual stimuli? And so what we needed first is actually a way to integrate our single-cell RNA sequencing data. So tons of people are using tons of different platforms, number of cells, algorithms, and we wanted to be able to use all of that data, especially as they become very public. And so the question that we asked is, what can your iPhone teach you about data integration? And so this is the work of an incredibly creative computer science student in my lab, Bryan, who said, this sounds a lot like how integrating single-cell RNA sequencing data sounds a lot like how when you take a panoramic image with your iPhone, how the iPhone integrates those different pieces of data. And so what you want to be able to do is focus on the cell types that overlap and integrate those, and then actually, keep the ones that don't map to any other images separate. And so that's what we did. And so if you think about it, we have an algorithm that's called panoramic stitching. It's available on GitHub. And so what you might imagine when you do this algorithm with a set of images, given a random collection of images, you need to first find those that overlap and stitch them together. Then second, you want to exclude the photos that don't belong to any panorama. And then once you have your matches, you can begin to assemble them. And so you can imagine an algorithm that does something quite similar if you had single-cell RNA sequencing data. And so the idea here is that instead of images, we have single-cell RNA sequencing data. Instead of pixels, we have single cells. And instead of RGB values, we have gene expression. What we want to be able to do is actually see, can we actually use this algorithm and apply it to single-cell RNA sequencing data? So what we did is we took every public data set we could download. And so at this point, I think our kind of mega data set is about 1.5 million cells. And what I want to draw your attention to is just the results. And so if we actually do this without any kind of panoramic stitching correction, what you find is you actually get a lot of batch effects. So if you actually just take the different 10x PBMC data sets, they actually don't really cluster exactly together, like your monocytes from one data set are over here and the monocytes from another PBMC data set are over here. You get macrophages kind of falling in different kind of t-SNE space. Really strikingly, when we actually apply our panoramic stitching correction, what you actually find is now similar cell types are actually falling exactly on top of each other in these data sets. And if we compare this to kind of the kind of industry standard that exists right now, which is this method called ScRAN, mutual nearest neighbors, you can see that it's not really an interpretable result. All the different cell types are kind of mapping on top of one another. And so the question is can we now actually think about going back to our single-cell RNA sequencing data sets that we're generating in the non-human primate and thinking about our in vitro data and actually try to integrate those. And just to give you a flavor for what that looks like, we can actually take the different markers that we're generating that kind of indicate where in the ontogeny the different cell types came from, what are the different markers of kind of differentiation cues. So we actually have a number of markers that can tell you if you were differentiated with M-CSF versus GM-CSF. We can actually begin to look at where those cells fall in the different granuloma populations. And this is really exciting because now we can actually say, if we see that a particular population of macrophages in a granuloma are associated with bacterial control or associated with any other phenotype that we're interested in, we can actually begin to extend our algorithms to improve our kind of in vitro experimental design. So to try to figure out how do we actually generate these cells in vitro, how do we test their functions. So to conclude, what I hope to have shown you today is that we can actually use single-cell RNA sequencing to really begin to unpack kind of the complex ontogeny of different cell kind of states and transitions that are happening in TB granulomas. Second, that inflammatory macrophages, at least in our hands, are appearing to adopt a semi-stable phenotype, at least using the markers that we have kind of at our disposal right now. And one of the things that I didn't really touch on, but one of the things that we're really excited about thinking about, given the kind of the subsequent follow-up studies we've done, is we were actually thinking about using protein expression in these macrophages to really kind of tell and try to unpack the cellular history, because if you think about it, these cells are no longer dividing, they're kind of turning on the expression of one protein, they're turning on the expression of another protein, and so we can begin to recover kind of cellular histories, and that as we move into this new era where we continue to move into eras where we have lots and lots of data, I think it's really important to think about how we actually use algorithms to really empower our understanding. So with that, I will just close. So I really want to just thank the people who did the work in my lab, Brian He, Chris, and Josh for the macrophage biology. All of this computational analysis has been done in collaboration with a really great computer scientist and mathematician at MIT, Bonnie Berger, my postdoc advisor, Sarah Fortune, and the Flynn and the Shalick labs for the non-human primate work. And thank you for your attention.
Hi. Yeah, that's very nice. I have a technical question on your single cell stitching together. So you compare that with the MNN algorithm, right? So, and you assume different cell types in different origins to be the same. So how do you exclude that the same cell types actually have different gene expression profiles a little bit? Do you neglect that? And how do you distinguish between batch effect and actually a subtle biological shift within one cell type?
Yeah, so that's a really good question. So the question is how do we distinguish kind of cell type identification versus maybe subtleties and difference in state? And I think what is essential to be able to define differences in state is to have kind of really robust differential gene expression. So if we actually take macrophages where we had plus minus LPS and then do the stitching algorithm, what you'll find is that you'll have your unstimulated macrophages and your stimulated macrophages kind of in a similar geography, but slightly shifted. And so I think there, what we're starting to be able to really ask the question of is how much difference in gene expression is necessary to move the population over. But the thing that we also then subsequently have been trying to figure out is how to really robustly better get at this question of batch effects, because I think these experiments with the public data sets are very easy to try to think about aligning, but in thinking about how do you take different macrophages in different kind of biological states and try to stitch them together, and maybe you don't actually want them all fall. That's kind of how we're improving the algorithms right now, but we're still working on that.
Brian, that's beautiful work. So the plasticity, like you observed for the macrophages in the bacterial infection. So for many of us studying tumor immunology, we are wondering whether, it's the same thing in the tumor microenvironment. There's tumor-associated macrophages, M1, M2 phenotype people talking about. So could you relate like what you're finding over here and shed some light?
Yeah, no, I've been thinking a lot about tumor-associated macrophages because I actually think that, this like some people like to think as tumor-associated macrophages being bad, right? And I think that so my working model, if I had to pontificate, is that similar to this spectrum that I described here, I think that as the macrophages probably see too much inflammatory stimuli, they move into this M1 star state. And so what we know kind of anecdotally and what we're working on right now is what we think is happening is that the cells are kind of accumulating either metabolic damage or insults that are really kind of pushing them into this extreme that's kind of almost abortive so that they can't be brought back. So I think that there's actually a very, very, very obvious connection between the tumor-associated macrophages and these phenotypes that we're seeing, for sure. That's a great question.
Thank you.